Well, thank you everyone for joining this panel discussion today. I'm very excited to have a fantastic group of researchers and the team from the movie and people that have organized this event for National Postdoc Appreciation Week for everyone to enjoy and to appreciate how postdocs are contributing to science. And with that, future postdocs as well, all the graduate students and people that are entering the field. So thank you very much, everyone, for joining. This webinar is going to be recorded and will be accessible on the Gladstone Web YouTube channel and as well on the UCSF NPower 2020 website if you want to rewatch it or to share the webinar. Another thing that I want to mention is that you can ask questions on the chat and we will collect these questions. And at the end of the panel discussion, we'll select 2 or 3 questions, depending on how much time we have. And then we will ask this question to the panel. So feel free to ask the questions in the chat and we'll get to that. So before we get to our panelists, I would like to acknowledge the people that have made this event possible. So I would like to invite on stage, I mean, virtually, Neil and Gaia from respectively Gladstone Institute and UCSF. I would like also to introduce Sudha from National Postdoc Association. And I would like to introduce the movie director, Bill Haney, that have worked together with Sudha and all this team to make possible the virtual screening of the movie and the panel discussion that we're having today. Sudha, do you want to say a word about how this partnership came together, and then we'll hear a word from Bill about how the movie came together?
Sure, Joel, thank you. Welcome everyone, and it's a real honor to get to be on the virtual stage, as Joel said, with all of you. I'm Sudha Krishnamurthy. I am the Director of Postdoctoral Affairs at Gladstone Institutes, and me and my counterpart here, Gabriela Monsalve, have been organizing a variety of events for our postdocs across our institutions. So this event came as an opportunity which Uncommon Productions reached out to me a few months ago about potentially screening the movie Breakthrough about Jim Allison's life. Being a researcher myself and from MD Anderson, this was something I was very passionate about. And I thought this would be a great story for our early career scientists, for our graduate students and our postdocs, to see the perseverance and what we can achieve. This resonated a lot with many of us, with our students, our postdocs, especially considering the pandemic and all that we are going through. So started from there, we reached out to Uncommon Productions if we would consider doing this as a wider event across a national channel, with the partnership with UCSF and National Postdoctoral Association. And they were very helpful, and now here we are. Now I'd like to invite Bill Haney, who's the director of the movie of Uncommon Productions, to tell us a little bit about how the movie came about.
Thank you, Sudha. Thank you, Joel. It's a privilege to be with all of you today, and a special treat to be with folks who are, as postdocs, kind of launching the next stage of their career in the world of science. The story that we filmed, Jim Allison's extraordinary personal story, and the story of the development of the first immuno-oncology drug that came out of his early thinking, and early thinking by Max Krummel and others, to me is an especially important tale about the world of science generally, and is rooted in work that he was doing in college and as a postdoc himself. And I hope that serves to some extent as an inspiration to you, as indeed it has to me. The world in general and the country in particular are facing challenging times. COVID is difficult. We see racial tensions in a really challenging place. Partisanship is on the rise, and a debate about the Constitution is, in front of the news. And it's easy to be drawn into this kind of vortex of darkness, with the information before us. But there's wonderful things happening too. And a lot of while there's darkness in Washington, there's sunshine coming out of the labs and the scientific institutions of the world, generally in the United States in particular. And in that sense, Jim's tale is a particularly inspiring one. His commitment to doing something extraordinary for cancer patients worldwide is, if subconsciously, rooted in his own personal challenges with the death of his mom from cancer when he was a youngster. And while this is a path that could have led to personal tragedy long term for him, he managed to turn this into a crucible of kind of extraordinary commitment to science and the inspiration that it can offer to the world at large. So, at least for me, the story of Jim's magical journey is a story about the creativity that is rooted in the hard sciences in a way that many audiences wouldn't necessarily think. If you ask the average American or person around the world to name 5 most creative figures they know, they will name authors and poets and playwrights and actors and film directors and musicians. How many will name a scientist? But indeed, the sciences can be the most creative of all professions. Celebrated in the Constitution itself, one of the few rights actually in the Constitution is the right of creative scientists to the results of their work, the inventions. Even rights so celebrated as free speech and freedom of religion aren't in the US Constitution, they're in the Bill of Rights that comes later. The Constitution itself captures one central right, which is the right of creative people to the intellectual property that results from their work. Patents and copyrights, and in this case, the kind of intellectual power of science. So I hope you find in Jim's tale, or found to the extent you've seen the film to date, and the link will leave up for a while longer so you get a chance if you haven't, this story of inspiration and creativity. You'll see a story of leadership, leadership that's about something bigger than themselves and their own enrichment and their own power and stepping on the throats of other people. That's not leadership. Leadership is about a purpose bigger than yourself, sacrificing time and time again in service of something more important than you, in this case, science, and more touching and soulful than you, in this case, cancer patients. And Jim's leadership and the gifted team of collaborators that he built together, Max is here with you today, are examples of what leaders do. They help inspire teams, they work collaboratively, they do it in service of something bigger and more important than themselves, and they do it when it's a struggle, and they do it when it's joyful. And in that sense, Jim's amazing tale has been a source of inspiration for me, and I hope for you. And the final thing I want to say is that while for many people this tale is a tale of Jim and Max and the scientists who worked with him initially, at least for me, it's also an extraordinary story about his glorious wife, Pam. Her own journey from Guyana through Queens and to Boston for medical school and PhD as an extraordinary woman and a woman of color, to the clinic where she helped pioneer trial designs that would launch the immuno-oncology revolution the world over, and to the partnership she and Jim have formed both personally and professionally to take immuno-oncology to the next step. And for those of you who are, I think hopefully you will find her story as inspiring as Jim's. And I encourage you to kind of go online and find more information out about her work and the work that they've done together since the completion of this story. And with that, let me thank you for having me and wish you all the very best.
Thank you so much, Bill, and thank you for making such an inspirational movie and for sharing these words with us. I would like to now move to introducing our panelists. And I will start with myself. I am Dr. Joël Babdor. I am a postdoctoral researcher at UCSF in the Spitzer lab, and my research uses multi-omic and systems approach to understand patient-specific factors that contribute to immunotherapy responsiveness in cancer and other clinical contexts. And now I have the pleasure to welcome and introduce the other panelists, Dr. Lisa Butterfield. She is Vice President of Research and Development at the Parker Institute for Cancer Immunotherapy, and also an adjunct professor of microbiology and immunology at the University of California, San Francisco. Her research focuses on cancer vaccines and cellular therapies. It's also my pleasure to introduce Dr. Justin Akien, he is a Parker Institute Fellow and a Principal Investigator in the Department of Microbiology and Immunology at UCSF, where his lab is developing a gene editing platform to enhance CAR T cells and CAR NK cells functions in hematological and solid tumors. We also have the pleasure to have Dr. Max Krummel, professor in the Department of Pathology at UCSF, His graduate work in Jim's lab has been a cornerstone of the discovery of CTLA4 as a target for checkpoint blockade. Max's lab has made an important number of contributions to our understanding of the role of myeloid cells in tumor immune response. And Max has created the Baker ImmunoX initiative at UCSF that uses multi-omic and computational approaches to improve our understanding of the immune system so we can harness it and to fight multiple diseases that are threatening human health. It is my pleasure also to introduce Dr. Avery Posey, Assistant Professor of Pharmacology at UPenn. His lab has produced fantastic work at the crossroads of the glycoimmunology, the cancer surfaceome, and T-cell therapies. His lab is now developing innovative strategies using CAR T-cells to target and eradicate cancer cells. I would like to introduce Dr. XingXing Zang, professor at the Department of Microbiology and Immunology at Albert Einstein College of Medicine. Xing has been part of Jim's lab and has advanced the field with the discovery of numerous immune checkpoints and is studying how they regulate the immune system. And his prolific lab is focusing on developing novel strategies to target these proteins to improve cancer therapy. And I would like to introduce now Dr. Pam Sharma, professor of the Department of Genitourinary Medical Oncology, Division of Cancer Medicine at MD Anderson. Her work in cancer immunotherapy has produced a countless translational and clinical contribution on the therapeutic strategies of harnessing the immune system by targeting tumor antigens. And last but not least, Dr. Jim Allison, who has dedicated his life and work to understand how to use the immune system to eradicate cancer. Jim is responsible for the discovery of the T-cell receptor and the associated CTLA4 checkpoint, which targeting allows to unleash the activity of cytotoxic T cells against cancer cells. His fight to make the discovery and a treatment available for cancer patients has led unanimous recognition in the field and has pioneered what it is today the most promising therapeutic approach to cure cancer. And therefore, he has received the Nobel Prize in Physiology and Medicine in 2018. Thank you all for being part of this discussion today. I'll start right away with the question about music. I know we want to talk about science, but let's start with that. Jim, you have an incredible harmonica skill. And there is this misconception in science that if you want to make it in science, you have to forget about all the distractions. And I would like you to share about what playing music and vibing represents to you and how having another passion outside of science has contributed to your success.
Well, I've been messing around with music since I was a kid. You know, I always liked music, many different kinds of music. I picked up the harmonica when I was a kid, and then gradually taught myself how to play it. Originally playing blues, but then when I was a postdoc at Scripps, I ran into some guys who they were Texas expats living in San Diego, and they had formed a country and western band called Clay Blaker and a Texas honky tonk band. And so I fell into playing with them. I played twice a week at this club called the Stingery.
As a postdoc? Yeah,
as a postdoc. And, because for the rest of the time I was working. Many times I went directly from the lab, to the club to play. And usually those would go on till 3 o'clock or so, and I'd still end up being at work the next morning. Unfortunately, with the Wednesday mornings, I had to give blood. I was the type donor for the HLA-9. I was the HLA-9 type. Gave the cells for the HLA typing laboratory. So, sometimes I hadn't slept, had too many beers, and had to give blood first thing in the morning when I got to work. That was the roughest part of it. But anyway, then later on, while I was there, I had the good fortune one time to actually go meet Willie Nelson and get him to come to talent night at the Stingery when he had a night off. But anyway, then that blossomed. So now I sit in with him every now and then. In fact, the last time I was in public really was March the 6th when I sat in with Willie at the Houston Rodeo. It's a lot of fun. But I mean, what everybody knows about though is a band called the Checkpoints that Lisa Butterfield actually participates in most of the time. It's all scientists and physicians. All of us do immuno-oncology, hence the name of the band. We do a lot
That's awesome.
we do a lot of SITC gigs for the Society for the Immunotherapy of Cancer and other kinds of benefit things to raise money for cancer research, cancer training and stuff. But mostly we just have a good time. And Lisa sings with us a lot. And in a way, I don't think, in some ways it's not that different from science, because in science, you try to bring together people with disparate interests and different kinds of specialties and get them to work together, because it's like a band. It's like putting together a lot of different people that work together. And after a while, you get to, I'll just finish somebody's sentences, but know where to think, or if there's a question, you can sort of anticipate it. And so, Max and XingXing can talk about that, the approach. But it's not like that in the band. Everybody's doing their thing, and then when you get together, and it's really clicking nicely. It's just like such a good feeling. That everybody, you can communicate with a nod, or whatever.
Yeah.
The player, he's getting right. I mean, he'll have the feeling, well, we should have a harmonica solo here, and he'll just sort of nod at me, and so I know next time I have to pick it up, and then I nod it to the keyboard player or something, and it's
so a band is like a scientific team, basically.
Yeah, it's a lot like that. Everybody has their own thing that they do, but it's the sum of all of it that makes the story at the end of the day, that makes the song.
I see. I'll turn to other panelists, and I'll ask Lisa, what is the hobby that you have that help you and you to your well-being and to be successful as a whole?
Well, with the highlight of my year is any checkpoints gig. That's absolutely the best. The other thing I do in my spare time is historical reenactment. And I enjoy transporting to another time and place and thinking about what life would have been like then. And that involves singing and dancing and wearing authentic costumes. And
wow.
It's very transporting. It really separates you from the day to day. And as a woman, I always think about, if I'd been born in 1890 or 1570, I'd have just been married at 15, washing clothes, and I would not be my own person working in the lab and contributing to something greater than myself in any way possible. So it's also makes me glad that I'm here now and able to do something that I choose to do. So that's a transporting hobby that takes you away.
Thank you for sharing this. I'm going to do a quick run, and the other are just going to tell me the name of the hobby to see, what are different hobby. I'll start with Justin. What's your hobby?
Well, mainly cycling and running.
Okay.
I think a lot about research when I do that.
Ah, that's good. Max, what's your hobby?
I second Justin's. I think you got to move through space if you're sitting at the bench all the time. But I think you also need something that is creative. Jim's got music. Sometimes I build furniture because you actually have something at the end of it all that's actually tangible, as opposed to science where you make a graph and no one really cares except the people on this call.
I see. Thanks for sharing. XingXing, what's your hobby?
One of my hobbies is reading Chinese Kung Fu novels. Because, this way, I don't have, like Jim, I don't have talent for playing music equipment, but I read Chinese Kung Fu novels, which actually is some kind of combination of history with human imagination. So, which allow me my mind traveling in different world.
Awesome. And we'll finish with Avery and then Pam.
Sure. I appreciate public radio. So I do a lot of listening to public radio, especially during tissue culture, that and karaoke. And probably as of late, gaming, video gaming.
Oh, nice. What do you sing, Avery?
What do I sing?
Yeah.
I sing along.
Nice. That's a good answer. Pam, what about you?
I think reading. I love getting immersed in a good book and feeling like I'm transported somewhere through the novel and stories of people and inspirational stories how people have overcome. I thoroughly enjoyed Michelle Obama's book, Becoming. Just a great story to read, and I could read it a billion times. And lately, for the last 2 weeks, my latest hobby has been our new puppy. So it's a full-time job.
It's a full-time job. I agree. Thank you very much, everyone, for sharing these hobbies. I think it's very important that to normalize that, you know, being in science is, yes, a lot of dedication, but I think it's important to have other passions outside of science, and it brings us back to science. It gives us more strength to come back and sometimes help us to come with new ideas and a fresh mind. I'll move to the next question, and now we're gonna start to talk about science, and we'll probably do this really, you know, in a summarized way as people have seen the movie. Jim and maybe Max, do you want to briefly tell us what the discovery, what was the biological significance of your discovery?
How we got there?
You're muted, Jim.
I've always been interested in what regulates T cells activity, and after the T cell receptor became clear there were other things. There was second, you know, the so-called co-stimulatory second signaling, and Jane Gross in our lab and a few others showed that the molecule that did that and seemed to be the most important molecule like that, it's actually required to act, in addition to T cell receptor to activate naive cells, was a molecule called CD28. And we had cloned CD28 and you know, made antibodies to it. But then there was a sequence that came out of the gene bank that looked a lot like it, particularly the extracellular domain. And so that suggested maybe a link in function. It was called CTLA4, and nobody knew anything about it except that there was a cDNA that was not found in resting cells and was found not just in CTLs from which it was cloned. It's the fourth molecule in this particular cloning, hence the clever name. But it's also on CD8s. So CTLA4 is a misnomer. But CD4 is still, it's also on CD4s. It's on all T cells basically, but only after activation. And then it was shown that it bound to the same ligand or bound to these molecules called B7 that CD28 bound to. And it was shown by another lab that CTLA4 bound to the same ones. And then the question was, what did it do? And so Max had the great luck to get this, you know, his thesis was, you know, what does it do? And so why don't you pick it up from there, Max?
Yeah, it didn't seem like great luck at the time. I think it took, you know, this is a molecule that likes to go inside of T cells. And so making an antibody against it was really non-trivial. A lot of the standard ways that were, you know, we were trying in the lab didn't work. But I think, you know, to your question, going back to your question for the non-immunologists, you know, coming into this period, we knew that the immune system could be tolerant and we knew it could be super active. And sometimes we'd want it to be active and sometimes we wanted it to be tolerant. And what we found was a way to dial it. Right, so CTLA4 and before a little bit CD28 were tractable ways that you could dial it up or dial it down. So like in a nutshell, like for a non-immunologist, you could make the immune system was like harnessed. It was yours.
That's an awesome way to summarize it. Thank you so much, Max. I'm going to turn to people that haven't been necessarily part of the work, but that their work must have been, I imagine, influenced by the impact of this work. I'm going to ask Avery, Lisa, and then Justin, what this discovery has, how this discovery has impacted your work and also the field around. Let's start with Avery.
Yeah, we think every day about how the immune system is suppressed by tumors and how to overcome that. And in the work that we do with genetically engineered T cell therapies, we're trying to reproduce what we see in the successful patients who've been treated with checkpoint blockades. We're trying to generate that with single cell infusion and reproduce that every time. And so we think constantly about what CTLA4 and PD-1 what they're doing in our T-cell therapies and how we can alter that, and both through using some simple genetic approaches, knockout, and then also some of the more complex genetic approaches that UCSF is known for. Is knockout of CTLA-4, knockout of PD-1 the same as antibody blockade of CTLA-4 and PD-1? And how do we take that forward?
Thanks for sharing this. Lisa, what about you? How this discovery has impacted your work and the field around you?
I was already doing human translational cancer immunotherapy, working on cancer vaccines and going to meetings. And every year the meeting was the same. There'd be great mouse data on some promising approach. And then there'd be a human trial and there'd be nothing. But then there'd be, another mouse experiment and we'd, get excited. We'd go back to the lab and we'd try again. So when the data from IPI, clinical data, started to come in, there was just a buzz. And the clinicians were talking in the hallway and talking about responses. And I think it energized everyone. It realized it was, finally there's a thing we did in mice that translated to humans. And I think that told all of us that keep it up, because, what you do, if you keep at it and think about it differently or right, or keep collaborating, it may impact, and so we've seen sort of a series of things come forward. So I think it energized the whole field.
It has been the lights at the end of the tunnel, right? And we're still in the tunnel, but the light is there. Justin, how did this discovery affected your work and your career?
Well, since I'm working in the same field and Avery and also Lisa, I can only echo what they said, where really the comprehension of what activates and inhibits the T cells and how can we control that and repurpose this science to better engineering has been fundamental to our work. And yes, we're thinking about it every day. And I think if I would add something, it's really the way that he's been against the flow and nobody really trusting his science is really, I think, matter a lot for us when sometimes we feel that we're in the wrong path, and just keep pushing to really understand what it is. And finally, how many trainee and now famous scientists have been trained with him or contributing to the field is something that I have an enormous respect for. So it's only a hope for the future. So, yeah.
So you're saying not only the discovery has impacted the way that you work, but also the inspiration from how he's been fighting, right? And yeah.
Definitely.
I can relate to that. Definitely, absolutely. I'm gonna ask Pam and XingXing a similar question, but a little bit twisted. What is the next frontier in the field of immunotherapy? Let's start with Pam.
So I think the way I would look at it now is that we are at the tip of the iceberg, right? Immune checkpoint therapy certainly shows us what's possible for the immune system to do in terms of treating cancer and possibly other diseases as we become more familiar with how other diseases are affected by the immune system, and not just autoimmune diseases. I mean, I think things like ALS and, lots of neuroimmunology spaces are opening up now. So we're definitely at the tip of the iceberg. Now trying to understand, as Lisa pointed out, what's happening in patients becomes very important. So the data so far, Jim's lab produced a lot of preclinical data that was then translated into the clinic, but how the human immune response evolves and why some patients are responding while others are not. What's the tumor microenvironment looking at? How does the immune response play with the tumor microenvironment? What are tumor mutations doing? Those are all things that we're still understanding. And then how do we look at the compensatory pathways, because the immune system is so dynamic that over time there are other pathways that get regulated, and when they're regulated and in which microenvironment, whether it's a bone lesion or a liver lesion or the brain, they are all regulated differently. So how do we come up with rational combination strategies based on this understanding that we need to get to?
Thank you so much. XingXing, can you share about what's the next frontier?
So the way I see it, I think, of course, the immunotherapy hold great pleasure. Currently, we only achieve a small part of it. So my own opinion, I think there's a couple different ways to maximize. One is discovery of some new immune checkpoint pathways, which my lab have been done. We discovered some new immune checkpoint pathways. Some is only in the human, some shared by human and the mouse. And then second, I think it just Pam and Lisa point out how we can move this basic study to the clinical trial, because if you don't do clinical trial, you never know that the final results. So the way my lab is doing is that we have a collaborator with some established joint companies and establish our own startup companies, and then we try to move these drugs to the clinical trial, because as an academic lab alone, there's no way we can move into clinical trial. We don't have the resource and don't have enough funding to do that. We have to work with venture capital, with a drug industry. So the third, I think, is a combination therapy, and then we should expand from cancer, but autoimmune disease and even other disease. I think currently most of the work has been done as a focus on the cancer, but clearly I think it holds great pressure, great hope for other diseases as well.
Thank you so much for sharing this. I think it's a great segue into the next section of questions that are related to research and perseverance. And in substance, you're saying that it is important to connect with the industry and the pharmaceutical industry in particular to make the partnership that matters. So I want to turn to Jim and come back a little bit on the movie where it's really well shown how it took 15 years of fighting, literally, to get your checkpoint blocking immunotherapy approved by the FDA and recognized as the drug to cure cancer. And your grit and determination to bring your discovery to the patient has been instrumental to overcome skepticism and resistance, and in all the fields, right? In academia, in pharmaceutical industry, in the medical and regulatory institutions, to get the treatment to the patient. It has been such a bumpy road, right? And your fight has been, opening probably more reactiveness from the industry side. My question is, Jim, do you think that going to this direction, that these institutions should in some way become more open to new ideas? And if so, what's the way for this institution, the pharmaceutical industry, academia, and the regulatory institutions to become more open to these new ideas?
Well, I think it's the way to do it. I mean, it's not easy. I mean, in the case of immunotherapy, as Lisa said, there were so many failures that everybody just put it in the trash can, and said it'll never work. I mean, that's what I heard so many times. Immunotherapy has never worked, it never will. I heard that from the drug companies, and I heard it from my colleagues, that were doing small molecules and stuff. First of all, I didn't get into it. I didn't start working on this stuff with the idea of curing cancer. I just wanted to know how T cells worked really at the bottom, at the end of it. It's just when, after Max's work and our work showing that you could manipulate this, and the data that we generated in mice, was just so compelling, because it wasn't one kind of cancer or one instance or anything. It was potentially applicable to every kind of cancer, because you're treating the whole immune system itself, not any particular one. So you didn't need a vaccine, you didn't need to identify anything. And but in the data, by the time we got to the point where I tried to convince companies, we had tons and tons of data with many different strains of mice, many different kinds of cancer. Essentially always successful, if not by itself, with something like radiation or a vaccine in some cases. Yet when we went to established companies, the scientists sometimes would get excited, but then they'd get up to higher and higher in the organization to people that had more seniority, had been around. They'd say, "Nah, it's never worked. It'll never work." Or issues about a patent of something that isn't widely apparent to people. Said Bristol Myers Squibb had actually made an antibody to CTLA-4, but they got the biology wrong. They thought it was another on molecule, and so the applications they had for it were completely different. So when we came along, we patented the pathway, the purpose, not the antibody. We patented the pathway for enhancing cancer. But the business guys, a couple of times we got all the way through the scientists at a couple of companies. Then the lawyers said, "No, BMS has already got a patent." I said, "But it's wrong. It's just wrong. The biology is the opposite. You'd never use their antibody to treat cancer." They said, "Doesn't matter. They got more lawyers than most of the companies that do scientists, So between those two things, it was finally we ran out of big companies to talk to. And I won't go through all of it, but finally, at a Gordon conference, I was desperate and having a beer with an old friend of mine named Alan Corman, who worked for a little biotech.
Metarex.
Well, at that time it was, but it became Metarex. They had a technology which was just outstanding. It was, the mice that had the immunoglobulin genes replaced with humans, so they could make a human antibody just straight away. No need to humanize it. It's fully human antibody, ready to go into patients. And so, but they didn't have an application. So anyway, we got together. And they said, "Well, we'll take a chance," and so that's where ipilimumab came from. But that took about 5 years from the time we published the first paper to really get to the point where it was even being taken seriously enough to try to make a reagent for humans. But then when it went into phase 1 trials, the first trial, which is, phase 1 is safety. You don't even think about a clinical signal. 3 out of 14 patients had objective responses, and at least one of them is still alive, and that was from 2001. So she's almost 20 years out now. After one injection of the antibody. Anyway, that got a lot of attention from people, and finally then it took off. But still, the first phase 3 that was done was Pfizer, and they shut their whole program down but their phase 3 was, I won't go into the details, but it was a poorly designed, poorly thought about thing, and main thing was they didn't wait long enough. And Metarex had gotten linked up with BMS, and they almost had to shut it down too, because of advisors after Pfizer did. But so even when we got there, and it looked like I was just, oh God, after all this. So we're in 2007, I think, at the time, and they almost dropped it, but they kept going, and finally in 2010, they got a successful phase 3 trial. But it took all that time, including one where it looked like it was going to die right at the end of the process. So it was amazing. Yeah, about 15 years.
A bumpy road. On the same theme, I would like to turn to Max. And, I think resilience and overcoming resistance, setbacks, failure is a prime skill in research. And on that, you've spent 4 years of your PhD trying to develop this anti-CTLA-4 antibody approach. And you actually got scooped, but then you turned this into a tremendous success. Can you share how you bounce back? How do we, and in general, as scientists, how do we make it to shift and to bounce back after such hard, setbacks?
Yeah, I think this is the lab story, and we just heard from Jim about the idea of maintaining perseverance, and taking the idea out in the world. But for probably the postdocs on this, the more salient problems are how to deal with pain and suffering and setback. That well, for a little bit of perspective, we don't have a monopoly on that in science. This happens to everybody in all their pursuits, right? In all life, it's there. So don't think that this is something special, but we do get more of it than most people, right? Because we're trying to do things that no one's ever done before. So, we see a lot of setback, we see a lot of hardship. And I think the thing that probably characterizes people do really well in lab, and maybe you kind of have to decide whether you're this person or not, is one that enjoys playing. And you're like that you like playing with ideas, and you like playing with things and breaking them and fixing them and breaking them and fixing them, breaking them, fixing them, over and over, and then that doesn't bother you, right? It's like kids in a sandbox, the sand falls over and they build it again, they build it differently, and it strikes me that's kind of like what we do, and if you don't love that, if you don't like doing that, I mean, you're given one of the things I think is also sometimes is lost, but if you look from space. We have you have people that build out their garages with power tools to do stuff, and they'll spend all their extra money, and then they spend the weekend in there, and they might have like a $2,000 drill, and they're like, oh, it's cool, I can whatever mill a thing. I got like a $400,000 fax machine out there the government gave to my lab, right? I mean, I got the coolest tools in the whole world to play with, right? I mean, I have the coolest tools. And everybody in this call, we have access to that. And so, take perspective sometimes and say, hey, no one else is going to be able to do this stuff. Like, no one else. I mean, there's very few labs in the world that has it. So, I mean, I think there's a perspective thing on the motivation thing, right? You look at it and you say, I'm really unlucky that these things didn't work out. But you're also doing something that maybe only you could do because of where you are. So, for graduate students, that might be and postdocs, that might be worthwhile. I'm not saying you can change your mind, right? If you're ever down and out, you're just down and out.
Right. Thank you so much for sharing this. And I like the metaphor of the sandcastle. And I think, it looks very much like what we're doing on a daily basis. And having these cool tools as well that bring some excitement is also part of the motivation and how we bounce back. I would like to turn to Avery. Do you have a story of a major struggle and how you went out of this? I
have major struggles all the time, but I don't have a good story for this session, so maybe we skip to another question.
That's good. Justin, what about you?
Well, I mean, I think I had the same story that many people get where, I was a grad student, been working for 3 years on a project and got scooped and get scooped so much they were not even answering the questions I was asking, but the 5 questions after that. And so I was a little bit depressed, but then I work on another project, but eventually I start working back on this one, being inspired by the paper I've been scooped by. And as a postdoc, I then wrote a paper that I scooped the team that scooped me as a PhD. And eventually we became friends. And so I think, there's no such thing really as being scooped. I think you should be inspired by the paper that are scooping you because they may be helpful.
Yeah, that's beautiful. Yeah. I have myself a similar story. In 2 years of my PhD were focusing on trying to produce some molecular biology constructions to capture a particular endosome compartment, and all the approaches failed. And so 2 years later, I'm actually completely shifting, and I'm turning to cell biology approaches, and I learned from scratch confocal microscopy, and I could actually see and dissect these endosomes even better than I thought I would do it before. So I think it's all about shifting, keep going, taking perspective. I think it's very important in science, and I'm sure a lot of people that are watching us can relate to that as well. Thank you so much for sharing this. I will move to the next question, and about mentorship. In the documentary, Jim, you said that the main thing that you look into someone that would join your lab is curiosity. And I think it's really an important skill. And a lot of people are thinking you have to be smart or you have to be intelligent. And those notions are, not so clear, right? But curiosity is something that we can sense, right? Can you share a little bit about how you see curiosity in someone?
Oh, okay. Yeah. Well, I said that because there are a lot of smart people, and there are a lot of people who know a lot of things. There are people that know a lot more than I do about immunology and everything. But if you really are more interested in what people don't know, and you're on the edge, and it's sort of the same thing that Max was talking about. You want to know something that is not known. You want to be the first one to really know something. you're curious, how does this work? But, you just really want to understand it, and preferably something that nobody else knows, and so that's I think the reward, and the thing that makes it so hard, but that's the big reward, especially if it's something important. Hopefully it's something important. But that's it. I want the people who are, I wonder if, I wonder whether, and
Right.
That's one of the things that I loved in my lab, and it happens not always, but in that particular time, and Max can attest to this, I loved coming into the lab. People would be putting their heads together. What about this? What do you think about this? And, little arguments, not really arguments, but discussions. And that's what makes it fun too. That's sort of where almost the band thing comes in.
Yeah.
But everybody's curious, and you're curious about what the other guy thinks too. And you all chip in.
Nice. I'm going to turn to Lisa. And so about mentorship, what have you experienced in your personal story that you have seen as a major positive way of mentoring people? And then I'm going to turn to a couple of other people.
I guess what I learned during my PhD struggle was how not to be a mentor, because that guy had no idea. But what worked for me is being put in an environment where the only thing that limited me was me. No one was telling me not to do an experiment. No one was telling me I couldn't do it, that they knew the answer, there was no point. So being put in that environment, so that's what I try to create for my people is two things. One, just it's my job to get the money, to get the systems, get you the cells, get you access to equipment so that you can just do what it is you want to do. You can ask the questions you want to ask. And then we've talked about the perseverance and the resilience. And I think the only thing that balances that is the excitement over the data. And so if people are just, plugging away and working and then they go home, that's not going to do it. That's not going to balance the perseverance and the resilience that you need. So you've got to draw the energy for that from an overwhelming excitement about the data. When it works, when you've got something new, when you think you've got a new mechanism, it's nothing like that in the whole world. So when you've got that feeds and supports you through everything else. So those are the things I've learned.
That's beautiful. Thank you so much. Yeah, and this mentoring style that you're talking about is really something that I think is very important, and how it brings the trainees to the next level, having the field open for them to play, and yeah, that's beautiful. Now I'm going to turn to the last question, as time is running pretty fast, and I'm going to start with Jim. In 1981, you delivered a speech at the House of the State of Texas to prevent a bill that was planning to prohibit teaching of evolution in public school and replace it with creationism. You stepped out of the lab, and your advocacy had a major impact in making sure that the bill didn't get through. Today, in a very acute way, science is still being challenged, and relative, to climate change or to COVID, either, and among other things. I think, there is a misconception that scientists better keep focusing on their lab and their research. But, I would like to hear your opinion about what is the place of the scientist in our society, and what should we do, right? And I want to quote here John Lewis that, talk about getting in good trouble. And I think that's something that you've done as well. So yeah, can you share about that?
Well, I think you have to get involved. I mean, what was going on in Texas was with creationism. I didn't really try to argue the points of, much of anything, except I just challenged them to use their creationism to, one of the things I mentioned was how do you use your creationism to understand how a cancer escapes the immune system, or a bacteria becomes resistant to antibiotics, or how do you develop a drug to do something? I said, you can't do that with creationism. It's crap and it's useless. And so that's basically what won the day. I didn't try to debate the fossil record or any of the tricks that they play to get you off point. But today it's in a lot of ways even worse, because because what's being challenged now is just whether that's worth anything, you know, or it's all political, you know, whether there even is such a thing as facts now. I mean, we're all about facts, right? I mean, if there's no facts, we're screwed. We've been wasting our whole lives doing something for no reason, you know. You got to have the belief that there's some objective realities and they're true for everybody, you know, and nobody has the right to get in the way of that. And it you know, what we're seeing now is just so frightening, and it's killing people right now, the degree it's gone to, by all these lies about the COVID virus and how it spread, and, you know, the people that
doesn't exist.
Just, yeah, it doesn't exist, and masks don't work, and it's just and then, you know, Trump now saying that he's not going to necessarily listen to the approval of the FDA. If they make it too harsh, he's going to step in and get it approved anyway, even if they don't have efficacy data, you know. And it's just, you know, it's something that just have to be, well, I mean, I was 25, I think, when I did that. And you just have to be alert all the time, because there are people out there that'll do that. And so we've got to do a much better job of educating science, you know, and what it is important, you know. I think it was accepted by, you know, 3/4 of the population at least until recently, and now I wonder, you know, I just wonder. And Pam and I together made 4 trips to Congress last year, you know, talking about the importance of funding research and everything. And luckily, most of the people we talked to, regardless of party, were strong supporters of it. So I just don't understand what's going on now. But
we have to be involved.
It's dangerous, and we have to be involved.
We have to advocate for, and we have to be involved at every level. We should not just be sitting in our labs and doing nothing. That would just be wrong. We are the ones that have to step up and speak and say what's wrong when we hear politicians, you know, blatantly disregarding facts and science. That just, we cannot allow that to happen, because then we're not really upholding the oath that we've taken as scientists. In a way, the ethical and moral implications of a scientist is that you are listening to the facts, and as a physician, you are doing the best for your patients. So those things have to still be upheld, regardless of the partisan politics, and we have to speak up.
I firmly believe, you know, Max was talking about how much fun we have with our toys and stuff, and, you know, it's the public by and large that buys those toys. So I think we have a right to do science to pay them back for that fun, and we can't let a bunch of know-nothings get in the way of that.
Yeah, share those toys. I guess use those toys for good, and share what we've learned with these toys, but also sharing those toys as well, right? I think, you know, there is a lot of discussions about diversity, equity, and inclusion in academia in particular, and I think, you know, a lot of people are getting into the fight right now and have understood that we need to do something about this. I'm going to turn to Max quickly, and Max, do you want to share what you think, you know, scientists should do regarding to that?
Well, I guess I could start by riffing on you know, in a jazz way on what Jim just said, is that you know, the idea that we shouldn't act on these things is defeatist. I think there's this idea that, you know, our place is in the lab. That's how the public sees us. Here's our place. This is where we belong. Don't like mess with us. And I think that the idea that we can't have an influence outside of our lab using logic and like good common sense and argue back like we do in lab with people when they come forward and say that's not the fact, that's not the truth. And I think there's also an element to which we're pretty good at looking at things where people say we've always thought that, and then saying, well, you should think differently because look, here's the facts. And we're faced with a situation where people are saying we've always done it this way. We think the problem is solved this way. We think it's fixed and decided how we're going to go. This is kind of our area. This is where we say no. Here's facts. Here's a way to think about it. And I think this is true when we think about diversity issues in academia or outside. The idea that we can't have any influence on things outside or even inside about how we nurture people from different backgrounds, bring immigrants in, all the things that are really near and dear to us because they represent the diversity of our thought. The idea that we can't influence those because what we're good at is pipetting. No, it's not what we're good at. We pipette because we're good at thinking about stuff and like devising, new ways of thinking it. And I think that's what's important in these arguments is to say, don't just think your world is around the pipette tip or the flow cytometer. It's about ideas.
Thank you so much for sharing that. Avery, is there a fight that you think scientists should get into?
I think, this goes along with the first question that you asked about what are our outside hobbies. We're not just scientists. We are all inhabitants of some place. And I think everyone on this call is this country. And so we should be a part of it. We should advocate for us seeing the best part of that country. So for me, I'm most passionate about seeing more people who look like me in science and fighting against some of the what I think are excuses, similar to what I think the Wells Fargo CEO said and got a pushback for recently about lack of representation being due to a lack of talent in the people who we seek to see in representation. And that is just not the way that I see the world. Not the way that I see the world.
These common misconceptions that keep being told and that are simply untrue.
They are told in science as well. They're also mentioned in science, and I don't think that the way that we see our science is due to lack of trying. That's not it.
Yeah, I agree with that. There's a lot of work on that side. Lisa, what do you think? Where do scientists should get into?
What we should all be able to do is answer the questions and be everything we can be and contribute in every way we can contribute and contribute ideas equally. And when we find ourselves in positions where we're not heard, we don't have a seat at the table, our opinions aren't considered, our ideas are not seen as equivalent, then we're not gonna help people. We're not gonna cure cancer patients. We're not gonna answer scientific questions. We're not going to solve these mysteries in a way that helps until everybody's ideas are all around that table and all heard. And then instead of 10 people that all look the same competing over 2 ideas, we're going to have 10 ideas and everyone working together to explore those and figure out what the truth is. So I think these are all fights that inhibit the science. And so it is our business.
Thank you, Lisa. Justin, what do you think?
Well, I think it's, as a scientist, we're lucky to be trained as scientists. And so I think it's a duty to be involved. And I keep fighting sometimes with people in my family that do not see the differences between a fact, an opinion, and a hypothesis. And I think it's really important to explain to them why exactly where this discussion is not, the fact that the planet is getting warmer is a fact. And the reason why it's getting warmer is something we can discuss about, but not the fact that it is getting warmer. And I think that, we have to be involved at every level. And so I totally agree with everything that's been said. Yeah.
Thank you. Shane, what do you think, is the last fight that scientists would get into?
Yeah,
fight that should get into, sorry.
Yeah, so I think, traditionally, some people think scientists just only focus on their research in the lab. However, I think now more and more people understand that we should speak out. If some idea statement is wrong, then we should speak out and tell the people, in the whole society. This is not right, or at least we should discuss it under the science guidelines. So then more and more people will understand. This actually I think would have a positive feedback, because if the whole society understands better of the science, they will more support us to do research, right? So I think this is actually also the scientific community would get the benefit as well in the long run.
Thank you so much for sharing this. All those words are so inspirational, and I agree, Al. We have to fight and we have to get into this fight, because I think this is or us scientists have this responsibility to transform the world and make it a better place. Thank you so much. I'm going to turn now to two questions that have been asked by the people in the public. And so I'm going to read the questions from Juan Perez that has asked, the CTLA-4 story is a great success tale of the importance of perseverance and stubbornness in the sciences. But do you think the attitude also risks getting into biting a dead horse? That's the first time I hear that, but I think I get it. Where scientists get stuck in projects with no good end, how do you teach your trainees about telling apart which projects are worth persevering or not? Where is the balance, right? I think it's a good question.
It's a very good question, because what it comes down to is you've got to know when to quit. I've had to tell many and had disagreements with many postdocs over the years that say, well, this one more experiment will get me over. I said, but you've done all this, and I think we're not, maybe technology won't allow us to answer that or something, but you're going to keep going. You can do this. If there's one more experiment and it's just this little bit different, it's probably not going to get you there, so you pick something else, but it's really difficult to find that point when you're gonna, you're gonna let go and move on to something else. It's almost like, when do you have the publishable unit,
the perfect paper.
Don't try to make it, and if you realize that you don't know what the next thing you're going to do is, probably it's a good idea not to do it. If you're not sure it's going to get you an answer that's beyond what you already know, then you wasted your time. But it's I don't know, it's
a fine line.
Yeah,
it's a fine line.
I think it's intuition. It's something that takes experience, but it still remains somewhat intuitive, I think. But you just got to sit down with some people sometimes and say, you're killing yourself over that. And it's what I would say, it's when you can't get an answer, as long as you're learning new stuff along the way, and you can think of what, but you should be thinking, it's like a good chess player, I guess, although I'm not a chess player. I played dominoes. You should be thinking 5 plays down, your next 5 plays. And if you can't, then maybe you're at an end, I guess is a way to put it. That's a really great indicator. I like that. Thank you so much. The next question is also for Jim, I think. Can you describe the exact moment when you realized the power of CTLA-4 inhibition, or when you knew it was going to be a cure for cancer? Was it a particular experiment or a moment outside the lab? I guess we can ask, both Jim and probably Max as well, right? Because I think, well, for me, I won't tell the whole story. Max is tired of hearing it, but it's when I wanted the experiment to be done double blind. And so that one guy set up the experiment. He showed me some data, and I said, this is unbelievable. Because I was expecting to slow tumors, and they just went away. There were just none. He said they grew well and they went away. And I said, wow. And so he set it up again, and I read them. I went in and measured them myself, and it was really depressing because for a while they were all the same. I had these cages of mice, and they were all the same. And then, but I got so depressed, I skipped a couple of readings, which you should never do, by the way. I would kick my students if they did it. But anyway, I skipped them. And when I came back, then there was a discernible, even just looking at the cage, there was a discernible difference. And then, it was clear. One group, all the tumors grew and killed the mice. The other groups, they all went away. Every one of them. No statistics, no nothing. It was just so obvious. And, but that's gave me the clue. But then we had to expand it to more different types of cancers. So the very impressive result, right? The one that you're sure about. Yeah, I don't know if I've get anything like it yet, but I hope I'll have a chance to someday. I would like to thank everyone. Yes, go ahead. After a while, though, then you have to do it again and again. That's when sometimes they say, well, I did it, I got that result. Well, you still got to repeat it. That's when it gets tedious. Thank you so much for sharing that. I'd like to thank everyone for being part of this discussion. I've learned a lot and had an awesome time, and I hope that everyone
that has been watching us has also had a great time. An hour is fast, and I would want to keep going and discussing with all these wonderful panelists that we have here, but I'll let everyone go back to our sciences and our life as well. I would like to thank everyone for organizing both the projection of the movie or the viewing of the movie and this fantastic discussion that we've had today. And I wish everyone a good weekend and great science for the years to come. Thank you, everyone.
Stay safe.
Thanks, everyone. Bye.
Thank you. Bye-bye.